To perform a multiple sequence alignment, we need to enter into a tool called Clustal Omega. So I am going to Google search and typing Clustal Omega. By entering as Clustal Omega, here you can observe the Clustal Omega is a multiple sequence alignment program that based upon HMM, that is hidden Markov model methods, and generally this database requires, this server requires 3 or more sequences. If you are having only 2 sequences, you can prefer for pairwise alignment. So to perform this, I am going to download 3 protein sequences. So to download the protein sequences, I am moving on to NCBI. As we are all aware, NCBI stands for National Center for Biotechnological Information. So in that NCBI database, rather saying database, I will claim it as data warehouse. And the data warehouse is none other than a database of many databases. So in such, the NCBI is having a database called protein for protein sequences. So I am preferring that. And I am going to search for a simple peptide, defensin. So I gave defensin and click search. Now let me take 3 different species. So defensin from Drosophila, defensin from Sultana, and defensin from Homo sapiens. So I took a protein from 3 different organisms. Once I have chosen, so there is an option to download the FASTA file. Go to send to, click file and choose FASTA and click create file. So in such a way, I'll get my FASTA sequences of 3 organisms. To have a perfect difference, what I'm going to do, instead of I'm removing all the assertion ID and I'm putting my genus and species name with a small underscore. Once I done with that, I have same, I copied. I don't want to change any other options. Simply I'm going to give a title for my understanding, MSA of defensin. This is just an optional. It is not mandate. Submit and let us wait for the results. Yes, and one more important thing to note, your results will be available till the day 7. You can see the results now. Say 94 sequences among the 3 organisms. You can see how the results are aligned. So all this star refers to conserved sequences, like valine is conserved over this position, correct? Whereas the colon refers to similar. Methionine and valine belongs to hydrophobic residues, so they are looking similar. But the reason for GAMS, you might know that all the amino acids are differing physicochemical properties. So this is the way we have to perform multiple sequence alignment. Thank you for staying tuned us.